So our group focused on gene expression changes in governing extreme dehydration tolerance in an Antarctic insect. Before we focus on the study, I would like to give you some background information. It is essential for organisms living in arid climates to maintain water balance and cope with dehydration stress. Insects are easily dehydrated because of their small body size. There are different ways that these insects alleviate this problem, which include physiological mechanisms used to maintain water balance, and molecular mechanisms which regulate cell osmosis. Polar environments are the most difficult to cope with because the water is frozen and unavailable. Belgica antarctica is a polar arthropod that lives in Antarctica, and the ability to tolerate dehydration is important for this species. This species is able to undergo another form of dehydration, called cryoprotective dehydration, which is when a decrease in temperature creates a vapor pressure gradient that depresses the body fluid melting point, allowing larvae to remain unfrozen in low temperatures. The focus of the study included the use of RNA sequencing to measure mRNA changes in response to dehydration and cryoprotective dehydration of different species, and these responses were studied in B. antarctica with the Arctic arthropod M. arctica. The study yielded the result that there was a strong relationship between gene expression and end products during dehydration. In order to conduct the experiment, B. antarctica larvae were transferred from substrate in ice water and left at 4 degrees Celsius overnight to maintain body water content. The desiccation condition was exposed to 93% humidity at 4 degrees Celsius for 5 days. The cryoprotected dehydration condition had temperatures lowered from -0.6 to -3 degrees Celsius over 5 days with ice, then held at -3 degrees Celsius for 10 days.
The caterpillar fruit fly is adapted to desiccating environments as well. The similar responses in the Antarctic midge show that downregulation may be a commonality of xeric-adapted insects. There was, however, no similar response in the mosquito.
In order to combat the desiccation of insects in Antarctica, scientists observed the enrichment of several functional terms, specifically the GTPase enzymes commonly found in membrane trafficking, some of these being heat shock proteins and cellular chaperones. During this experiment, they noticed a regulation of autophagy. Autophagy is a catabolic process in which parts of the cytoplasm and organelles are sequestered into vesicles and digested in lysosomes. This process in turn conserves cellular macromolecules and energy during periods of stress and nutrient deprivation, in this case, desiccation. They identified 92 homologs of genes known to perform autophagy. The first piece of evidence needed would be an 11-fold upregulation of cestrin during desiccation. Cestrins are simply highly conserved genes that have an antioxidant function that help prevent cellular death. The second piece of evidence needed to support this claim is a significant upregulation of 6 autophagy-related signaling genes. The third piece of evidence is upregulation of 4 transcription factors. The fourth piece of evidence is a 3-fold upregulation of thread. And the final piece of evidence needed is an upregulation of proteasomal genes. This suggests that there's a crosstalk and cooperation between these distinct cellular recycling pathways. They hypothesize that the autophagy pathway serves an important protective function by limiting cell death and turnover of macromolecules during dehydration, especially during long Antarctic winters.
Functional categories downregulated during dehydration. Downregulation is the process by which a cell decreases the quantity of a cellular component, such as RNA or protein, in response to an external variable. This is similar to the term negative feedback that we have previously discussed in class. This study observed downregulation of ATP production and general metabolism. In particular, the study found that larvae of B. antarctica limits its oxygen uptake significantly in response to dehydration. Downregulation continues as we find that metabolic depression is found to be a common adaptation of dehydration-tolerant insects, the rationale being that it minimizes potential water loss from respiratory functions. This inference strongly supported by resulting data of gene expression, in which 25% of all genes responsible for metabolic processes shown downregulation under the stress of desiccation. Interestingly, though, and logically, the genes related to protein, lipid, and chitin metabolism showed an increase in expression. ATP generation was halted in response to desiccation. The majority of genes responsible for glycolysis synthesis displayed signs of downregulation. The coordinated shutdown of the metabolic processes within the larvae of B. antarctica is one of the key contributions to energy conservation of the overwintering arthropods.
The researchers used targeted GC-MS metabolomics to check the levels of gene expression in the compounds. What they found is that 32 of the 36 compounds, or roughly 89% of them, had significantly altered gene expression after desiccation and cryoprotection. Although changes induced by desiccation and cryoprotection were similar, each compound was unique due to hierarchical clustering, which is seen in the diagram below. Some of the metabolic responses to desiccation include lower levels of glycolytic intermediates, glucose 6-phosphate and fructose 6-phosphate, which indicate a downregulation of glycolysis genes, decreased levels of lactate, more citrate, which shows a deep downregulation of the citric acid cycle genes, increased levels of proline and glutamate, which is a precursor to proline, more osmoprotective polyols, and an upregulation of phosphoryl pyruvate and carboxykinase, which increases glucose production via gluconeogenesis.
The Arctic midge, the related species, shows very similar results to the Antarctic midge in relation to desiccation and cryoprotective dehydration. There were 1,280 putative 1-to-1 orthologs found between these two species. 4 were found in common that helped carbohydrate hydrolysis and a single peptidase. Although many dehydration-related genes are expressed in common, the profiles of the Antarctic midge and the Arctic columbolan were drastically different. The transcript signature is more dependent on the species than the form of the dehydration present. This shows a different path of evolution in similar environments between these two species.